My name is Rebecca. I'm the Associate Professor of Biology and the Director of Public Health Program here at Agnes Scott College. I've been at Agnes Scott for 6 years, and my background is in researching protein structure and function and how it relates to various diseases. I'm here to welcome you to this year's Scottie Book Month. We will be discussing The Immortal Life of Henrietta Lacks, a book written by Rebecca Skloot. I still remember the moment I first heard about HeLa cells. It was when I was taking my undergraduate molecular biology class. The professor spent some time talking about tissue culture and different cell lines, and then he introduced these cells called HeLa cells, and he told us that they were named after women. I believe he gave us the wrong name, Helen Lane. In that sense, my first experience with HeLa cells actually parallels that of the author, who also heard about these cells while taking a community college biology class. She became fascinated with this question of who is this woman and where did these cells really come from? HeLa cell line is one of the most important cell lines for tissue culture research in biology today. So I wanted to tell you a little bit about what tissue culture research is all about and why it is so important for modern biology. Biologists like to rely on model organisms such as fruit flies, mice, etc., to study various diseases and important questions in biology. But ultimately, we often want to know what happens in humans. And cell lines are a powerful tool that allow us to ask these questions without harming any individual human subject. So cell culture basically allows us to grow human cells in laboratory conditions and to expose those cells to various environments, such as drugs or potential pathogens causing diseases. HeLa cell line is very special because it is actually an immortal cell line. What that means is that it can be grown in laboratory for a very long condition, indefinitely. The cells keep on dividing from 1 cell to 2 cells to 4 cells, and they do not stop. That is very different from our normal cells, which can only divide a certain number of times before they die. So what scientists do in lab, they use HeLa cells and they grow them in media like this, in containers like this at appropriate temperatures. And what this allows them to do is to multiply the cells and to grow them and to observe them under conditions that they are interested in. Today, HeLa cells can actually be ordered from catalogs. In fact, just today I looked up in American Tissue Type Culture catalog, which is a standard catalog for ordering biological samples, and we can order HeLa cells for about $300 for an academic lab. So it's a very common line that's used by many researchers. It's circled around the world, and it actually had some important contributions. It made some important contributions to science. For me, some of the most fascinating ones were using HeLa cells to determine how many chromosomes we have. We all take it for granted that humans have 46 chromosomes. We learned that in the introductory biology class, but that was not known in the 50s. HeLa cells is where that observation was first made. Another incredibly important contribution was the use of HeLa cells in polio research. Thanks to HeLa cells, we were able to develop the polio vaccine and to help stop the spread of that awful disease. The list goes on, and it includes applications such as AIDS research and control of various other diseases. So how did HeLa cells come about? Henrietta Lacks was a patient at Johns Hopkins Hospital in the 50s. The hospital was segregated. It was one of the very few hospitals that would even admit Black patients at that time. Henrietta started showing symptoms a few months after giving birth to her 5th child, and she went in to get tested. It turned out she had cervical cancer. And as a part of her treatment, the doctor removed a part of her tissue without obtaining any kind of informed consent from Henrietta. She really didn't know what was going on. She knew she was in a hospital to receive treatment. She trusted the doctors. She hoped for the best. But little did she know what would become of that sample. The sample was taken to the lab, where a lab technician was diligently working on various samples received from many women that were treated for the same condition. The technician would take that sample and try to grow it in a petri plate. In the 50s, that was very difficult. They didn't know what kinds of chemicals to put in the petri plate to allow those cells to grow and replicate. And it was a bit of a trial and error. Most samples that the technician got did not grow. Henrietta's sample was different. The cells kept on dividing and dividing. And in fact, the technician did not believe it for a while, thinking, oh, they'll die just like any other cell line. Well, that did not happen. And from that, the first immortal cell line was born. In biology, the word immortal simply means that these cells can divide many times, infinitely many times, without stopping. So the conditions under which that sample was obtained were quite different from what a patient might experience today. I might add, it was routine back then for doctors to take samples from patients without informed consent. It just so happened that Henrietta was treated in the colored ward of Johns Hopkins Hospital, and that her cells were actually capable of growing. However, other samples were taken from patients of various races, and it was always done without consent. In fact, there was not any kind of standard practice for informing patients about risks or potential consequences of their act of donating a tissue sample or organ or anything of that sort. And in that sense, that is quite different from how samples are taken today. Despite problems with informed consent and lack of patient privacy, the author makes a point that the researchers and doctors involved did some good things, in fact. Back then, there was no legislation protecting patient privacy and sample donation practices. And in fact, they treated Henrietta the way they would treat any other woman. Treatment options were fairly limited at that point, but she did receive adequate medical care. The author also points out that the researcher who first grew the cells was not interested in making any kind of profit out of them. In fact, he circulated the cells to any other researcher all across the world who was interested in pursuing relevant questions in biology. And that kind of practice is to be admired, and it still happens today in science. For example, when scientists discover new genes or new sequences or new protein structures, it's pretty common to share that information with other researchers. And in that sense, what was done back in the 50s is admirable. Now we're going to give you a few questions to motivate your discussions of this book. On one level, The Immortal Life of Henrietta Lacks is a story about race and racism. How would this story be interpreted differently in 1951, the year of Henrietta's death? Are there any alternate interpretations today? Do you think the family should be compensated for what happened to Henrietta? What form should that compensation take? What would you expect if that was your family? A New York Times reviewer comments that the author, Rebecca Skloot, is a character in the story herself. How might the story be different if she were not there? What ethical principles were used to make the decisions regarding Henrietta's cells? What ethical principles do you believe should have been used to make those decisions? Thank you for joining us for this year's Scottie Book Month. I hope you enjoyed the discussions.
Greetings from Agnes Scott College. My name is Kim Vickers, class of '87, and I'm the Senior Director of Alumni Relations here at the college. Thank you again for participating in this year's Scottie Book Month with our faculty-led discussion by Srebrinka Robic in our Department of Biology. The day after we filmed this for Scottie Book Month, Srebrinka left for Croatia to get married. So here's a belated congratulations to Srebrinka. Please stay tuned for a special interview conducted by Harry Wistrand with alumna Tamika Strickland, class of '97. Tamika Strickland just received the Outstanding Young Alumna Award for her work in the medical field with women's health. Tamika had the unique opportunity of working with HeLa cells when she was a medical student at Morehouse School of Medicine. We hope you'll enjoy the interview. It lasts about 10 minutes. Thank you.
Hello, I'm Harry Wistrand, Professor of Biology Emeritus at Agnes Scott College. I'm here with Dr. Tamika Strickland, biology major from the class of 1997. We're going to be talking with Tamika about her knowledge of the HeLa cells. Before we begin our discussion, let's find out a little background from Tamika about herself.
Hi, so it's nice to be here today, and it's great seeing you again, Dr. Wistrand. To be really quick, when I finished Agnes Scott, I started medical school at Morehouse School of Medicine, and from there stayed with Morehouse School of Medicine and completed my residency program in obstetrics and gynecology at Grady Hospital. From there, I transitioned to Arizona, where I worked for 4 years on the Native American reservation, on the Navajo Native American reservation with the Indian Health Service. And from there, I transitioned to Tucson, where I now essentially practice obstetrics for a perinatal group in southern Arizona, and then have an evolving interest in international women's health.
Well, when did you first learn about HeLa cells and the story behind them?
You know, there's a physician, a GYN oncologist, that is a part of the Morehouse School of Medicine faculty, and he has been a mentor in my life now for almost 15 years. I first met him my first year in medical school, and then had a direct teaching connection with him as a resident during the OBGYN department. But about 15 years ago, in the mid-90s, he started what's called the HeLa Conference. That was actually my first time ever hearing the name HeLa and understanding and being able to make the connection to the greater story behind it, which is Henrietta Lacks. So he started this conference that's every year, and essentially it's in honor of Henrietta Lacks, and it's a conference that focuses typically on women's health and primarily with a focus on cancers involving women.
Yeah. Well, how has this knowledge affected your own work as an OBGYN?
In many ways, it certainly affects myself and the larger generation of medical students and young physicians from a perspective of ethics in science and in medicine. It's pretty standard of care for us to always approach patients with research, with any kind of study, or any kind of procedure with informed consent. And during the time of Henrietta Lacks, it was not the standard of care for any kind of study or procedure to be done on patients without that designated form that essentially puts together a legal right for the patient. So from that perspective, it's really, really important. Moral and ethics in medicine is just as important as the science behind it and breakthroughs and being able to move forward with technology. From another standpoint, as an OBGYN, women's cancer, you know, cervical cancer, breast cancer, all the cancers involving the reproductive tract are very important. And it's a lot of the innovations that evolved as a result of the technology used with the HeLa cell has been pivotal in us being able to treat women today. There's the Casci line, which is CASKI, that Dr. Patillo actually developed himself, was based off the model of the HeLa cell in terms of him being able to cultivate that line. And that cell line was actually pivotal in developing hydroxyurea, which we still use today for treating a lot of reproductive tract cancers, as well as and innovative treatments for sickle cell anemia as well. And so the treatment of cervical cancer, the treatment of so many other reproductive cancers has evolved and derived out of studies that were allowed through the HeLa cell.
Right. So it's been a very important contribution to medicine.
Absolutely.
What does the medical profession owe the Lacks family? Because they've got quite a tragic story in this book.
Absolutely. And it's been a long journey, obviously, that Rebecca Skloot tells so beautifully in her book. And even today, there's a Henrietta Lacks Foundation that Dr. Patillo actually sits on the board of, and donations from the book and from other members of the community goes towards a scholarship program to help support children and grandchildren that are a part of the lineage of Henrietta Lacks. But in terms of being able to establish some form of financial reimbursement from the billions of dollars that has been generated in the bioscience, molecular genetics, chemotherapy research arena, there's not been there hasn't been established any legal way in which the family can receive financial compensation. But obviously, the fact that her name is huge and important in terms of her impact upon the world. I don't think we mentioned earlier today that it was the biggest medical breakthrough that came from the HeLa cell was the development of the polio virus. And if it was not for that cell line, we'd still have complications and children dying from that disease. And so her legacy upon the world is huge, and that can never be taken away anymore, and that's important.
I found in reading this book, it was, as a biologist, had some good science in it, but it's also a book that's very readable for the general public. Even if you have no science background, just have an interest in understanding human stories and understanding how medicine operated back in the 1930s, 1940s, 1950s. You get quite a compelling story. Did you find the same thing?
Absolutely. Rebecca Skloot did an amazing job. She's an amazing creative writer. I think she has a master's in creative writing with a background in science and biology. And to read the book was very much a history lesson. It was a storytelling. It was beautifully put together in a way that reached the general population as well as scientists and physicians.
I was amazed at her persistence in getting to this story. I think a lot of people would have given up.
Right.
And her having to work with Dr. Patillo, I know he was very hesitant to talk about this at the beginning. Why was that, do you think?
It was important for him to protect the family, understanding what they've gone through before, and even the series of events that unfolded that disclosed the fact that the HeLa cells were in fact from Henrietta Lacks still all came from an element of not full disclosure. And so his primary concern at that point was making sure that the family was protected, and it was her persistence, meaning Rebecca Skloot's persistence, that eventually won him through and allowed an element of trust between him, Rebecca, and Henrietta.
So I think Tamika and I can recommend this book highly to anyone who is interested in history of medicine, how medicine treated different groups differently, and what the outcome of those things are. It's a very compelling story of a family. Thank you very much.
You're welcome. It's good to see you.
Good to see you too.